Okay, so maybe we can start. So it's my great pleasure to have Michael Young today coming from Uptown and Rockefeller. And we're actually in the house of 19 Washington Square North, which is the house of NYU Abu Dhabi. So what is NYU Abu Dhabi? But 16 years ago, I think President Sexton decided that New York was too small and not diverse enough, and we need to expand to other territory. So he managed to create NYU Abu Dhabi, where we have about 2,000 students from 120 nationalities who are studying there very hard, very competitive, very great campus. And as part of this campus, there's also an extension here in New York. And the goal here is to promote science and culture through the NYU Abu Dhabi Institute, which is basically housed here. And what we try to do is basically to promote the culture and then to have the people from neighborhood and from NYU to come here and to listen to great minds of our times. Okay, so there's scientists, there is artists, there's also kind of creation. Okay, so today we have a program called the Luminaries of Science, and so basically people who made big difference in science, and you will see Michael Young is one of those, and so Michael is from Florida, but I think he spent early part of his life in Texas, studied in Texas, and he did his PhD working on fruit flies, transposable these elements that jump from one place to another, repetitive elements. And then I think he made basically a big choice by going to Stanford to study with David Hockness, who was at the time the person who could walk not on the moon, but on chromosomes. Which means it was a time when molecular biology was evolving, and then we start being able to identify genes based on where they were located on the chromosome. So he was assigned to work on a small part of the chromosome, and then he basically was doing extremely well. And then he got a position at the Rockefeller University where basically he decided to do something very wild. I think to clone genes which were involved in behavior. And this was actually in the very late 70s, early 80s, where people didn't believe that gene could control behavior. And he's going to tell you the story about that. But basically what he did is that clone those genes and managed to show that indeed gene could control behavior, in this case, the clock behavior of flies. And what the beauty of the system is actually that it was a good example where studying these tiny fruit flies could actually illuminate, I speak like ChatGPT now, could illuminate the not only the clock in this small animal, but also in human, in any species basically where which is sensitive to light, which is basically us and all the other animals on Earth and all the plants and even the bacteria. And so basically this was an illustration that studying this model system could really allow us to understand what happened in much higher organisms. And the conclusion is basically we are all built on the same blueprints, and we use maybe tweak something to get different form of animals or plants, but we all use the same machinery. And so because of this discovery, and along with two other scientists, Michael Rosbach at Brandeis, and he actually received the Nobel Prize in 2017, yeah, and to basically illustrate how indeed working on those organisms could allow us to discover many, many things which are relevant to what we care about, human, but also the world in general. So, Michael, it's your turn, and then.
So, thanks, Claude. I don't know where you went. Oh, you took my seat. So it's, let's see, how do I get the full picture? That looks good. Okay. So it's a pleasure to be here. I'm going to elaborate on what Claude was saying about our studies of behavior. Circadian rhythmicity, or evidence for circadian rhythmicity, is found all around us. When I was a kid, growing up in Miami, We had a house next door. Actually, the kids would run through everybody's backyard looking at stuff. Nobody ever chased you off. And there was sort of a jungle in the house next door. And something that I noticed early on was there was a plant with these big white blooms. And when we'd play kick the can or capture the flag, whatever at night, I noticed that these blossoms were open, but they were never open during the day. And this struck me as really odd. You know, a plant that goes through cycles has a sort of behavior, it seemed. And I hadn't seen that in many years, but we have a place out in New Mexico where I ran across a similar phenomenon. I thought I'd take a picture of it and just show you what we found. So we're out on after my wife and I are out on an afternoon walk, and we go by this plant, which is called Mirabilis, and its pet name is Colorado 4 o'clock. And what you see here is a picture on the top of what we see at 2 p.m. or what we saw on this hike out at 2 p.m. And down here is what we saw when we came back at 4 p.m. You can see there's a huge difference in the healthy look of these flowers at 6 p.m. It's not Photoshop. This is the same plant. This same stick is there. So everything is legit. We hung around, we passed a number of these plants. We hung around one of these for a few minutes, again at 6 p.m. And we noticed this happening, which is you see this little moth. I don't know where he went. Here it is over here, pollinating these flowers. And here's another one coming up. And what we noticed over the next few minutes was the bush more or less lit up with these hawk moths. So this plant, it turns out, is almost uniquely pollinated by hawk moth. And that's a night active moth, evening and night active moth. And we're in an arid environment. Water's precious. It takes this plant has to pump water out of its leaves and roots in order to open those blooms. And what I suspect is happening here is it's in that arid environment, it's come up with a pattern using its circadian clock, its ability to time, to have those blooms presented only at times of day where there's an adaptive advantage to doing that, which would be when the pollinator's around. And so that being the case, this is, again, an example of the kind of thing you could run into if you keep your eyes open. A demonstration of circadian rhythms, this time in a plant. Now, in animals, these kinds of rhythms have been seen for many years. These are data from 1960. A scientist that's in University of South Carolina, Pat DeCourcy, is recorded the activity on a running wheel of a rodent, I think it was a flying squirrel, over 25 days. So each one of these lines is a different day in the life of this rodent. It's in a dark room, so it doesn't have any access to environmental cues about time of day. And every time it gets on its running wheel, it produces the graph of the is produced so that these deflections from that horizontal line appear. So you can see the deflections peeling out day after day in this 25-day record. And what's remarkable to me still about this rodent record is that you can predict within 2 or 3 minutes when the animal's going to get on the running wheel, when it's going to get off the running wheel. Again, for 25 days without any external information coming into that rodent. The other thing you'll notice is that it's not a vertical pattern. It's spread off to the left. So each day begins a little earlier, each day ends a little earlier. We don't have environmental cycles, even if we were missing some that were coming through, that would have that period. We have a perfect 24-hour period in our environment. So this altogether, everything you see here suggests very strongly that there's something internal that is being read out with this rhythmicity. Now, a number of years later, a scientist at University of Massachusetts Medical School, Bill Schwartz, did an experiment in which he gave a rodent access to radioactively labeled sugar that was in drinking water. And this accumulates according to the metabolic activity of cells in the brain or anywhere else in the animal. And what they looked at here during the middle of the day versus the middle of the night, and they looked for any kinds of differences that would be day-night differences, and they saw this, which is this area of the hypothalamus called the suprachiasmatic nucleus, SCN. It showed this robust rhythmicity in metabolic activity, glucose uptake, in these rodents. That was followed by experiments that led to surgical experiments. What happens if we ablate this region of the brain? And the scientists went on to eliminate that region of the brain, and they found that the locomotor activity rhythms that I showed you previously went away. So the animal behaved as if it was clockless. And perhaps more impressively, they found that an animal lesioned in that way could recover rhythmicity if they were given a transplant from a fetal animal of the same tissue. So transplanting that region of the hypothalamus from another animal could restore rhythmicity, which, tells you all kinds of things. It tells you that, yes, this is a little pacemaker of some sort. It must be producing a diffusible product that is released into this region of the brain and has the effect of conveying information about time of day. Now, what we wanted to know is that's a particularly intriguing set of findings. But what's going on in the cells that compose that bit of hypothalamus that's able to produce that kind of information about time of day? And so what we turned to was a study that took place at the California Institute of Technology in 1971. I was a new graduate student at the University of Texas, and I saw this paper by Ron Konopka and Seymour Benzer. Seymour Benzer was very well known at the time for work on bacteriophage, but he had moved into studying behavior using genetics in Drosophila. And he and his student, Ron Konopka, did what is illustrated here. They found 3 mutants. They used a chemical mutagenesis to create the mutants. And they isolated one mutant that was arrhythmic, had no rhythmicity, acted like an SCN ablation. Another mutant, which had a period of 19 hours, 19 instead of 24, fast-running clock. And the third that had a period of 28 hours, so a slow-running clock. So this is like what else could you get? Fast, slow clock, and no clock, all at one genetic locus. They did analyses of the location of these mutations. They all seem to map to the same functional gene. And so that led me as a graduate student to be interested in seeing if I could find out more about what this was genetically initially. So I was fortunate in working on this region, of the X chromosome of the fly, which is roughly where Konopka and Benzer had mapped this set of mutations. And these are these giant polythene chromosomes of Drosophila. I don't know, some of you may have seen pictures of these in textbooks or magazines. They're produced by multiple divisions of the same double helical DNA. So you have 1,000 strands per chromosome. So you can physically, you can microscopically look at the physical organization of the chromosome as it runs along its length. And it's, of course, characterized by these heavy folded DNA-rich regions, which at the time that paper came out, I was studying to try to understand how many genes can be packed into a Drosophila chromosome. Drosophila chromosome has enough DNA to code for 50,000 genes, and yet all the genetics says no, maybe 5,000 at the time. So I was interested in that kind of a question, but I became very interested in these new mutants that had been found at Caltech. And I realized that if their mutations mapped in this area, I might have tools to better locate that gene. And in fact, these are the tools that I had to work with. I had deficiencies that are shown here. I had duplications that are shown here, and I had a number of translocations. These are chromosomes that are broken. Information is not lost, but they're rearranged in some way so that you could identify the chromosome break and map it. And what I found was when I got Ron and Seymour's mutants, I found that they all mapped according to this set of rearrangements to a single space in the middle of the region where I happen to be working. So how lucky could you be? So this is the most important of those mutations for later work turned out to be this translocation, T14JC43, because it broke right there. And in fact, when I asked, was there a phenotype associated with that break, I found yes, there is a phenotype. It's a rhythmic. It has no rhythmicity. And it was the same gene that Seymour and Ron had identified. Now, the advantage of something like that is that when I did finally wind up in California learning to use molecular tools to isolate individual segments of DNA, you can find a break. At that time, you could find a break in a chromosome pretty easily. You would never find a point mutation. I couldn't find their mutants, but I could find this chromosome break, which would identify the gene. So this was in 1978, but a number of years later, when I was running a lab at Rockefeller, we managed to locate that gene, as did our colleagues at Brandeis, Michael Rosbash and Jeff Hall. But we were able to map it to a single transcription unit, and we were able to see that translocation had broken that transcription unit in a way that explained the phenotype. So that nailed the particular product being produced to that functional gene. Now, we did a number of additional mapping studies on this, but ultimately wanted to know, there weren't many clues from studying that one gene about how a clock works. So we had one gene in hand, and we had a lot of information about that gene, but we didn't have a lot of clues about how this system would emerge from the action of that one gene. So we decided to do what Ron and Seymour had done, but on larger scale, and began to screen for ourselves for new mutants that had altered clocks. So this, in contrast to those flower pictures I showed you before, this is Photoshop. But this is what 7,000 bottles of flies would look like, and the postdocs who did this work are back there in the shadows. But the way this experiment was conducted is each bottle has to be shaken twice a day to find out if there's a difference between numbers of flies that have hatched in the morning versus the evening, day after day. And what you're looking for are flies that are hatching all the time, so that they've lost their clocks. And one of those bottles gave us that phenotype, and that was a new gene, which we named timeless. Now, the thing about timeless was that as we began to study that gene further, we found that this new gene physically associated with the protein made by the first gene. So period makes an RNA and a protein. Timeless makes an RNA and a protein. And the two proteins interact. They physically associate, and that association is critical. If that association doesn't take place, the clock stops. And the reason the clock stops is because neither of those proteins can get into the nucleus to do their job. One of the proteins, the period protein, just disappeared if we didn't have the timeless protein. So the way this, I'll skip ahead now to tell you how that protein pair and other genes that we found in the same kind of screening way work together to produce a 24-hour oscillator. So you've got two genes, period and timeless, in the nucleus that have similar regulatory sequences associated with them. There's a pair of transcription factors that work together. I just label them C and B, but it's clock and cycle are their names. And those transcription factors bind to the per gene and independently to the timeless gene and turn them on. And so they make RNAs when activated in that way. The RNAs are transported out of the nucleus into the cytoplasm where they're translated into two proteins, the period protein and the timeless protein. But then two important steps in regulation occur. One is that there's a protein kinase that we found again in our screening called double time, which physically binds to the period protein when it's by itself and phosphorylates it, marking it for rapid degradation. So when per is not bound to its partner timeless, period is destroyed. In the meantime, timeless, we found, is also regulated in an important way. It interacts with another protein called cryptochrome, which is regulated by light. It's a novel photoreceptor that when in the presence of light, causes it to bind to timeless and to modify timeless so that timeless is now destroyed. So during the day, there's never any timeless because cryptochrome is constantly destroying timeless due to the presence of light. And of course, if there's no timeless, as I told you, double time will destroy all the period protein. So, so all day long, the RNAs are accumulating for these two proteins, but the protein production is sterile. When the lights go off, the two proteins can accumulate as soon as these period timeless dimers are able to be made. And timeless carries period into the nucleus, and period and timeless then bind to clock and cycle and drive them off of these genes so that they can no longer activate the production of more period and timeless protein. So all night long, period and timeless are shutting down these two genes, and during the day, these genes are free to be active because of the absence of period and timeless. And so we get a cycle that is driven by patterns of light and dark. The cycle will also, though, continue in constant darkness because of some of the regulatory properties that I just told you about, in addition to other post-transcriptional and post-translational regulation in the system. But it's sufficient to say that this will run at near 24 hours in constant darkness, but at precisely 24 hours in the presence of a 24-hour light-dark cycle. Now, you might say that's a pretty simple system. How does that control behavior or other aspects of 24-hour regulation? And the way that occurs is that that system winds up regulating hundreds of other genes within the chromosomes. So what this chart indicates from top to bottom, each row is a different gene. And each row, each gene in these rows is oscillating with high levels of product indicated in red. And so this is a 6-day experiment, and you see low, high going on and on for those 6 days of the experiment. And we've organized this, I think it's about 150 genes according to the phase of peak activity of each of those genes. So nearest neighbors have the closest patterns of expression with regard to time of day. So those near the top are most active at dawn. Those here are most active at noon. These are at dusk, midnight, and so forth. So what I think impresses me about this pattern is that you see that these are diagonal stripes that are running out here. They're not vertical, which means that as we move through this chart, we're seeing genes that are coming on and turning off at many different times of day. It's kind of like a music box where you've got, you're playing a melody over the course of those 24 hours. There's a time and a place for each one of these activities, and the particular arrangement of that expression pattern is determined by those clock genes that I mentioned previously. So this is the same thing I showed you on the prior slide, but I also wanted to compare that to what happens in 3 mutants. This is clock, period, and timeless. And this is in a light-dark cycle. Everything is in a light-dark cycle. And what you see is that the patterns, the stripy patterns, have gone away, even though it's a shorter duration experiments. There's no evidence of an organized, a time-based organized pattern. So what that indicates to me is that in spite of the fact that this must be a system that originally evolved as an adaptation to environmental cycles early in the Earth's history, we're now, 3 or 4 billion years away from those initial events. And we've interposed, the organisms have now a biological mechanism for tracking time and has uncoupled itself to some extent from the requirements of an environmental cycle, to the point that an environmental cycle alone will not produce rhythmicity in any of these ordinarily rhythmically driven genes. You have to have the interposition of that clock system, that set of clock genes, in order to get this rhythmicity, so that if we eliminate any one of those genes, we lose the ability to produce rhythms even in the presence of a strong environmental cycle. Now, this is work that was done in John Hoganish's lab at Cincinnati Children's Hospital a number of years ago. And what we've learned about mammals is that about half, probably a little more than half of the genes in the animal are, and in us, are regulated in a circadian fashion. So those clocks are regulating a huge component of our genomes. And if we're just as a pause to give some medically relevant information, the Hoganish group found that 56 of the 100 most often prescribed drugs in the US have rhythmic gene targets. Now, that's not particularly surprising. If half the genes are oscillating, then it's probable that half your drugs are going to hit an oscillating gene. But what is perhaps more important is that it's also understood that of the top 100 pharmaceuticals, of the top 100 drugs, half of those have a half-life of less than 6 hours. So all of these drugs have a half-life of less than 6 hours. So now imagine that you've got an oscillating target and you've got a drug that lasts, whose half-life is 6 hours. After 18 hours, most of that drug is gone. Do you want to take the drug in the morning, or do you want to take the drug in the evening? This, unfortunately, isn't put on your prescription. I think it should be on the label. For example, a statin. You're taking a statin, should you take it at night or you take it in the morning? Does your physician tell you when to take it? You should take it at night, because your liver is producing cholesterol late at night. And so that's when you want to inhibit cholesterol synthesis. If you take the pill in the morning, you're going to have to wait 18 or so hours for cholesterol synthesis to get underway. Now, fortunately, most of the statins have very long half-lives. They don't have 6-hour half-lives like these, but it's an example of the potential importance of having your medication regimen connected in some way to the target you're trying to hit. So a number of years ago, another kind of question was asked. It's not only that many genes are regulated by these clocks, but these clocks are found all over the body. So it's not just in the brain. It's in lung, liver, skeletal muscle. And ordinarily, all of those clocks are in agreement because you're active, you sleep, you eat. All of those things are closely coupled in time. They wanted to know what would happen if you produced, as shown here, a brain-body conflict where you give access to food at the wrong time of day relative to the activity cycles of a mouse in one study or a rat in the other study. So in both studies, what they did was that this is a nocturnal animal. They provided food for an hour or two in the middle of the day. And they found that the animal would develop a new routine where they would wake up. After a few days, they started waking up just prior. They anticipated the food delivery. They'd eat the food and they'd go back to sleep. So they'd wake up, eat the food, go back to sleep. But then at sunset, when the lights went off in the experiment, they'd get on their running wheel and they would be active just like unaffected animals. So they had this bizarre new pattern of activity. And when they looked at what was happening with all of those regulated genes in these different tissues, they found that whereas the brain, lung, liver, skeletal muscle all agreed with regard to time of day, in the animal that was free to be active, to sleep, and to eat, according to the combination of these features, that now the lung clocks, the liver clocks, the skeletal muscle clocks attained new phases with respect to one another and with respect to the light-dark cycle. The brain stayed on cue. It still followed the light-dark cycle. But food was changing the organization of these other peripheral clocks. And so we've got a desynchronization that's going on because of the disagreement between the timing of food ingestion and the time of the activity patterns of the rodent. The other thing I'll just point out is this reminds me a lot of jet lag. And I think this is what goes on with jet lag. I don't think it's just that you arrive at a distant location and it takes time for you to progressively move everything forward. I think you just scatter your clocks and it takes several days to get them back together. For example, the liver is among the slowest organs to finally get back to that new location's time zone. So I think, again, it's a desynchronization rather than a failure to move fast to get to the new location. So we wanted to know, are there long-term effects of having that kind of discord? And a related experiment that we did in Drosophila and fruit flies was to say, what happens if we have free access to food around the clock, 24 hours a day, versus flies that are only given access to food for half of that time, for 12 hours a day, and matching the 12 hours a day when food is available to the time when they're active. So you've got food when you're active. In one arm of the experiment, you've got food all the time in the other arm of the experiment. And so here's what happened in that experiment. The health span of the, I'll call it TRF for time-restricted feeding, those that were on food for only 12 hours a day. This is the survival curve for those flies, and this is the survival curve for the flies that were on ad lib food. And I think, I don't have the numbers. Yeah, I do. So we're looking at hundreds of flies here to generate the data that are shown. In contrast, if we do the same experiment but use a fly that's clockless, either because it's missing per or it's missing timeless, we lose that we lose the benefit of having that time restricted feeding. So once again, it's not sufficient to have an environmental input. You have to have both the clock and the environmental input to have an impact. In this case, the longer the flies are on time-restricted feeding, the better their survival curves or their health span curves. So those that are for their entire lives, adult lives, kept on time-restricted feeding, those have the best health span curves. Those that are on for the ALFs, or again the trailers, and those that are on shorter times perform less well than those that are on for the entire length of the experiment. So, and the other thing that we did was to look at, well, what if you don't start the TRF schedule until they're 30 days old? There's still a benefit. So you can see that there's a parallel for the first 30 or so days, but then they start to separate after the TRF, the time-restricted feeding, is put in place. So I guess it's never too late to start if you're a fly, and the longer you're exposed to this kind of a pattern, the better off you wind up being. The other thing that we see is underlying all of this is a huge difference in the number of oscillating genes and the amplitude of those oscillations. So for example, this is the oscillation of one of the core clock genes, the clock transcription factor. And in time-restricted feeding in the head, much higher amplitude oscillation than in the head of equivalent flies that are in ad lib feeding, constantly available. And this is a count of the number of cycling genes in the body under ad lib feeding or time-restricted feeding, and in the head under constant access to food or 12-hour access to food. So a tremendous change, a doubling in the number of rhythmic genes are seen with that treatment. Now, I want to move now to a human story because I think, as Claude mentioned, what we found in Drosophila has led us to understand what's going on in us as well. Most of the genes that I've been talking about are fully conserved in us. I'll show you that those components in a minute. But before doing that, I'll just point out that we began to study in coordination with a group at Weill Cornell Medical School a few years ago, patients that have delayed sleep phase disorder. So these are patients that have a persistent delay in the timing of the major sleep episodes. You can think of these as chronic night owls, and a lot of resistance to efforts to advance phase. So these are people that are so troubled by their inability to get to sleep before midnight, well past midnight, that they'll come into a clinic to try to get some help. And it's pretty common. Estimates are maybe roughly 5% of the population falls into this category. So we began to look at subjects that had come into the clinic with DSPD. And one of the first things we did was to give them an activity watch, which they could put on just like a regular watch. And we tell them to wear it all the time. It's waterproof. You take a shower with it, whatever. And then it's kind of like those locomotor, that running wheel experiment in rodents. They're just like a rodent on a running wheel. You see these deflections wherever they're active. And this is organized in a way so that the sleep time is sort of put in the middle of this graph, but you're looking at multiple days for a control individual and multiple days of what we call tau 11, an individual that I'll tell you more about in a minute. But this is a DSPD individual that came in with DSPD. And what they also do, in addition to having the activity watch, they have a log by their bedside, and they're supposed to enter the time they go to bed and put a mark. When they get up, they're supposed to make a mark, so that the red triangles indicate when they got into bed, the blue triangles when they got up the next morning. And you see that the control individuals very well behaved, a good block of sleep each night. They're going to bed a little bit before midnight, which is 24, and getting up, at a very reasonable time, maybe 8 o'clock in the morning in most of these cases. The tau 11 individual is all over the place. I mean, you can see that it's a very ragged record, but one of the things that stands out is that they get into bed very late. I mean, this is well after midnight, and in some cases approaching dawn, and getting out of bed in some cases approaching noon. So this is a pretty severe case of night owl syndrome, delayed sleep phase disorder. So there were several members of the family from which tau 11 came who helped us with a genotyping study in which we did what's called whole exome sequencing. We're looking at all the protein coding genes, we're sequencing those, and we're looking for things that suggest that those that show DSPD in the family, are there similarities that are not seen in those that don't show the same syndrome within the same family. And we found one gene that popped up, and that's a mutation in cryptochrome, Cry1. And the nature of the mutation is that genes are made up with blocks called exons separated by introns, and those blocks are the protein coding components of the gene, and those get stitched together to produce a messenger RNA that will produce a full protein. And what this individual carried was a single nucleotide change in what's called the splice donor site of exon 11. What that means is that that change prevents that bit of protein coding sequence from being melded with the next block of protein coding sequence downstream. And so what happens is the protein coding sequence from block 12 gets stuck onto block 10 instead of block 11. So you have a deficiency of 72 bases in the RNA, and of where have I got this written? Well, okay, anyway, this is the RNA, and this is the RNA from subject 11, and this is these are a series of control RNAs, and you'll see subject as 11, number 11 is the only subject to have an altered two forms of the RNA. So they've got one good allele, and they've got one allele that carries this mutation with the altered change in the structure of the RNA. And here's the protein from the same group. So both the RNA and the protein are present in two types in that individual, indicating that there is a functional deficiency in both the RNA and the protein that match expectation. So again, most of the clock components are shared between Drosophila and us. Clock and BMAL are identical to, or nearly identical to, clock and cycle in the fly. Once again, this time a period cryptochrome dimer instead of a period timeless dimer is responsible for inhibiting the action of clock and cycle. And instead of regulating timeless, it regulates cryptochrome. So the mechanism is much the same. Per cry complex is what's necessary to move into the nucleus and what's necessary for stabilization of the proteins. And with that set of modifications, what we wanted to look at was whether or not any of these features, especially those involving cryptochrome, was altered in cells from that individual, from skin cells from that individual. And I won't show you the raw data. I'll just show you what we, in structural form, what we found is that the mutation in cryptochrome produced a change in the way that cryptochrome and her interact with clock and BMAL. And they, in fact, become a hyper-repressor. They become a more powerful repressor, which is why we believe they have a change in the rhythm of their clocks. And structurally, what appears to be happening is that the tail end, the C-terminus of the protein, wraps around the body of cryptochrome. There's a site, a docking site for clock on the surface of cryptochrome, and ordinarily that tail wraps around and dynamically occludes that interaction point. So what we have in this mutation is a loss of exon 11, which is what normally would be sitting in front of that docking site. So now the docking site is always available for clock. So clock is not partially inhibited, and we get and so we believe that is what's responsible for its behavior as a stronger repressor. So then we found something that I thought was pretty intriguing, which is we wanted to know, where is this mutation found across different populations across the world? It's very abundant in Europeans, or at least non-Finnish Europeans. It's rarely found in East Asia, and it's found at lesser frequencies in other populations. But if we think of Europeans, the allele frequency is 0.6%. This is a dominant mutation. The carrier that we have been studying has only one copy of that mutation. So the carrier frequency is going to be twice that, which means about 1 in 75 of these individuals, of these Europeans, have the capacity to display that change in behavior, which is a pretty striking frequency. So we wanted to, given that should be something we should find more evidence for, we set up a collaboration with a group in Turkey at Bilkent University, a lab of Tayfun Osalek, who identified for us in a large population he'd been studying for obesity, a number of families that were carriers for this particular variation. And shown in all of those shown in black are carriers. Males and females are shown in the circles and squares. And those that are unaffected are shown as open circles or squares. And what we looked at was the mid-sleep point for each of these individuals, whether they showed delayed sleep phase disorder or not. And the non-carriers are plotted here on a 24-hour clock face, and the carriers are plotted here on the same 24-hour clock face. And what you see is that the non-carriers have a mid-sleep point at about 4:30 in the morning, which may sound familiar to many of you. And in contrast, the carriers' mid-sleep point is something like 6:30 in the morning. So a fairly significant phase shift in their preferred sleep phase. The other thing we found in the carriers was another phenotype, which is this fragmented phenotype. Those are individuals that sleep in fragments overnight and call their first nap their main sleep episode. And so those were scattered around the clock. So it's a more complicated phenotype than just a night owl phenotype. It's a fragmented phenotype as well. So let me just summarize all of this. So we've looked at several unrelated families where the presence of this mutation, which we'll call Cry1 delta 11, predicted delayed sleep phase disorder. And the penetrance and frequency of the allele suggests a broad contribution to DSPD worldwide, or at least in Europe. Since the mutations found to have multiple members in each family, then each of those cases, all of the cells in those individuals should have that mutation. So we would expect this to affect more than just their sleep pattern. It's going to be present in their liver, their muscles, throughout their body. The mutation Cry delta 11 shows enhanced binding to its targets, clock and BMAL, which are the transcription factors that drive expression. And so we think that that enhanced binding is reflective of a strengthened transcriptional inhibitor. So we think that inhibition has been ramped up because of this mutation. The competitive binding to clock and BMAL, that is, not only is it a strengthened interaction, but it largely replaces the interaction of the wild type. From the wild type chromosome, it largely replaces the interaction of that protein with the target. And I think we think that's a basis for inheritance as a dominant trait. And then finally, something I didn't show you is that if we put that mutant form of the gene into a wild type mouse cell or human fibroblasts that have from subjects that have no clock difference from no DSPD, we can produce DSPD effectively in those transfected cells. So that once again ensures us that this is responsible for the circadian effect that we see with that mutation. So let me just point out the people that did all this work over the years. So Alina Potke, who's now out at Caltech, did the work that I discussed on cryptochrome, the DSPD work, in collaboration with the group at Weill Cornell, Scott Campbell's group at Weill Cornell, and Tayfun Ozilek at Bilkent University. Let's see, Sophia Axelrod and Daniel Cabrera, and Zik and Wang did the work on how flies behave when they're placed on time-restricted feeding. So examine the effects of ad lib feeding versus time-restricted feeding. And it's just a little celebration from a number of years ago. So thank you very much. Appreciate it.